Great. Welcome, everybody, to the CompuCell Workshop Module 5.2.3. Our guest presenter today will be Abhisha Thayambath from North Carolina State University, and she is a doctoral student in Professor Giulio Belmonte's group. Her research focuses on mechanical transitions and development in embryonic cell populations. And without further ado, I will turn over the screen to Abitha. Her title is Start Small, a model for tissue-wide planar cell polarity without morphogens.
Thank you, Ellen. I would like to first thank the organizers for this opportunity. And I hope my screen is visible.
Yes.
Okay. So today I'm going to talk about a model for tissue-wide planar cell polarity without morphogens. So first I'll talk about the story of the paper, and then I would go to the implementation aspects. So planar cell polarity is an important patterning mechanism in both vertebrates and invertebrates, in which the cells polarize along the apical plane of the epithelia. So this is mostly found in body hair orientation, convergence and extension movements in vertebrates, etc. So essentially, this is a subcellular pattern of protein localization where certain proteins gets localized to the proximal or anterior sides of cells, and the other set of proteins gets localized to the distal or posterior sides. So this is operated according to the core PCP pathway, which acts across molecular, cellular, and tissue levels. At the molecular level, you have local feedback interactions between the proteins across cell-cell junctions, so that would stabilize intercellular complexes along a specific direction. And as a result, at the cellular level, each cell aligns with respect to its neighbors along a specific direction. And at the tissue level, it has been proposed that there is some sort of a global cue that would align the overall direction to proximal-distal axis. So there have been many candidates for this global cue, which is thought to provide the directionality for the global alignment, as well as ensure long-range coordination of PCP across the tissue. And two major candidates has been the fat dashes pathway, and the second is the Wnt signaling pathway. However, there are experiments which showed that the fat dashes mutations do not affect polarity in the Drosophila wing margins. And it has also been shown that the uniform expression of dashes also rescues polarization. And recently, there are two experiments done in Drosophila wing where they knocked out the Wnt ligands and gradients and found that disruptions in Wnt also does not disrupt PCP in Drosophila. So there is no experimental evidence as of now for a global cue that provides this large-scale coordination of polarity. And various mathematical and computational models have been proposed to to understand PCP. So there are mostly two classes of models: explicit signaling models, which consider the molecular level processes explicitly. And then there are phenomenological or reduced models, which basically capture the essential dynamical features of PCP. However, most of these models use some sort of a global cue in the form of a tissue-wide gradient or like a small bias that acts within each cell. And most of these models require some sort of a global cue to ensure large-scale coordination and to provide directionality for alignment. So in the context of the recent experiments where researchers did not find the role of Wnt ligands in PCP alignment, and as of now, there is no experimental evidence for a global cue. And in this context, we asked this question, that if there are other mechanisms that establish long-range PCP. And to answer this, we used the Cellular Potts model. We developed a model for PCP in this framework. So as you all know by now, in this modeling framework, you have a generalized cell, which is basically a collection of lattice sites. And this generalized cell, as Julia was talking this morning, could represent an actual biological cell or subcellular compartments like nucleus, cell membrane, and cytoplasm, or abstract regions within the cells like apical, central, or basal compartments. So then the properties and behaviors are encoded in the effective energy, and the system evolves by energy minimization. So the question is, why do we need a cellular Potts model for PCP? Because it's much easier to add other cell behaviors like cell growth, division, shape changes, and cell rearrangements. So in this framework, we are going to use cell compartments to represent the protein distribution within each cell. So as Julia was talking this morning, each cell can be considered as a cluster and with different compartments within them. And we are going to use this idea to represent the spatial localization of PCP proteins within each cell. So in our model, every cell has 4 domains: cytoplasm, proximal, distal, and lateral domains. And we start the simulations by placing random seed cells in the lattice and let them grow and relax their boundaries. Once this is done, we introduce the PCP domains within each cell. And at the beginning, these domains are introduced randomly within each cell, as in it's distributed randomly. And then over time, just by using the contact energy preferences of these different domains within each cell and between domains of neighboring cells, the system self-organizes. So the way this happens is by at cell-cell junctions, the proximal and distal proteins want to form intercellular complexes, right? So they have high affinity, and within each cell, they want to be spatially segregated. So they have low affinity. So just using these differential contact energy preferences, the system self-organizes. So now, in order to quantify the tissue polarization, we use this metric, which is essentially the average of the normalized distance between the proximal distal compartments. So basically, we are averaging the polarity vector of all the cells in the tissue and then normalizing it across, and taking the magnitude and then normalizing it. So we find that at the beginning, when all the compartments are uniformly mixed inside each cell, the tissue is not having any polarity. You can see that the orientation vectors for each cell are aligned along random directions. But in this case, as time progresses, you can see that all cells have their polarity aligned from left to right direction, and the global polarization is 1, calculated using the metric. Now, before I continue, we tested if the model is capable of reproducing the mutant PCP phenotypes. So we started with the frizzled mutant, which is basically a scenario where the distal proteins are absent in a small patch of tissue. So the way we implemented it was by removing the distal domains from a small patch of tissue, as you see here. And we found that as time progresses, the tissue that is distal to the mutant patch has misalignment. Its polarity is disrupted at the distal side of the mutant patch. Similarly, for Van Gogh mutants, the polarity of at the proximal side of the mutant patch is disrupted. So the way we implement this is, so Van Gogh mutant essentially has no proximal side proteins, and the mutant patch is fully populated by distal domains, and hence the polarity on the proximal side is disrupted. So having established that the model can successfully reproduce the mutant PCP phenotypes observed experimentally, we went ahead and tested the polarity for different boundary conditions. So we started with a periodic boundary condition, and where there is no preferred alignment direction for the cells, for the tissue as a whole, so it can align in any direction from 0 to 360. And a second condition where we have a left boundary orienting signal, which was implemented by adding a column of distal domains at the left side of the tissue, which would guide the first column of cells to align from left to right direction. And then we asked, if we increase the system size, how would the global polarization change? So we found that as we increase the total number of cells in the tissue, global polarization decreases for both periodic boundary and left boundary signal configuration. And when we look at the mean angle of tissue polarization, we found that for the largest system size that we examined, the mean angle is uniformly distributed for the periodic boundary, whereas for the left boundary signal configuration, we can see that it's distributed mostly from proximal to distal direction, but it's more dispersed around that axis for larger system sizes. So for the periodic boundary, for the largest system size that we examined, we found that there are local domains of local alignment, and swirling patterns emerge over time. And for the left boundary signal configuration, we found that cell rows closer to the boundary are aligned from proximal to distal direction, but as you move away from the boundary, this alignment is gradually lost. So clearly, a boundary-orienting signal alone is not sufficient to generate long-range polarity. So then this effect becomes worse as we increase the system size along the x direction. Basically, if we add more and more cells further away from the boundary, you can see that the global polarization decreases to maybe 30% or so. So then the question is, how would the cells be, how would a tissue so large can have global alignment? So we hypothesized that maybe cells establish polarity in a small domain and then expand the domain while conserving alignment. So the way we did this was by starting with a small patch of tissue that is already polarized with the help of left boundary signal as well. So it's already aligned along proximal to distal direction. And then we let the cells at the boundary grow and divide. So they grow by maintaining the relative proportions of proximal and distal domains within each cell. And then as they divide, they lose all sort of polarity information. So once they divide, the proximal and distal domains are equally inherited by both the daughter cells. And during that time, these compartments are uniformly distributed inside the daughter cells. So the polarity is transiently lost. And then they look at their neighbors and re-establish polarity. So this has been found experimentally as well. So using this method, we found that when we look at the global polarization as the number of cells in the system increases, we found that it's pretty robust. So you do get about 90% polarization even when the number of cells in the tissue are increasing, as compared to a scenario where you have a fixed sized tissue that's not proliferating. But the problem was that when we look at the mean angle of tissue polarization, we found that it's not primarily aligned along proximal to distal direction in this proliferating tissue. We found that it's aligned along plus minus 45 degrees. And then we thought, okay, instead of dividing, growing and dividing the cells that are only at the front boundary, what if we let all cells divide, but the time at which they divide are chosen randomly. So we randomly choose the cells and let them grow and divide within the tissue. And we found that this mechanism is pretty robust at maintaining the alignment along proximal to distal direction. The only the important thing here is to note about the time scales. So every cell, when it divides, when it grows, should not disrupt the polarity of its neighbors. So the time scale of growth should be chosen such that the polarity of neighbors are not disrupted. And once it divides, it should have sufficient time to re-establish the polarity before it starts growing again. So by calibrating these two time scales, we found that this proliferation mechanism, where we let all cells in the tissue grow and divide, is sufficient to generate tissue-wide polarity. So the global polarization stays consistently higher at more than 90%, and the mean angle of tissue polarization is also aligned along the proximal-distal axis. So most importantly, this mechanism helps achieve polarization much faster than starting PCP signaling all at once, and it works in the absence of a long-range global morphogen. The next question that we looked at was the mechanisms of polarization in individual cells. So it has been found experimentally that whether single cells are able to establish PCP asymmetry is an organism-dependent problem. So recent experiments suggest that in Drosophila, individual cells, even if you prevent them from interacting with its neighbors, can establish PCP asymmetry, whereas in mouse, you need information from neighbors to spatially segregate the proximal and distal proteins within them. So we tested both these mechanisms within our model. I'll come to the details of how we implement this later, but essentially we found that when we do this, the results are qualitatively the same. So for both periodic boundary conditions and left boundary condition configurations, we find that as we increase the number of cells in the system, global polarization decreases. And once we introduce cell proliferation, the polarization stays consistently at 90% or so. But what we found interesting is that the cell non-autonomous mechanism is producing consistently higher levels of global polarization for both the boundary conditions. So we think that this could be because in the cell autonomous mechanism, there is one additional constraint on within each cell, which could be restricting it from achieving higher levels of polarization. So the next question we asked is about the question of emergence versus maintenance of polarity. So we started with a system that is already polarized along proximal to distal direction. So as you see here, and we compared that with a system that started unpolarized and is gradually polarizing over time. Interestingly, we found that the global polarization of this system decreases over time. And when we look at the mean angle of tissue polarization, which started along proximal to distal axis, starts showing the variance. So you can see that local domains inside the tissue starts misaligning. And then we asked, okay, what if we start with a tissue that's perfectly hexagonally packed? And then we found that the global polarization stays consistently at whatever value we started. So we conclude that the ability of a large tissue to keep its orientation depends on cell packing. So to summarize, local interactions between proximal and distal proteins can lead to local self-organization. And we found that a boundary signal can guide the alignment along a particular direction, but it's still short range. That's not sufficient to establish polarity in a large tissue. And in the context of recent experimental work in Drosophila wing, which suggests that wind gradients are not necessary for global PCP alignment, we propose this mechanism where local boundary orienting signal along with cell proliferation is sufficient to achieve global alignment. And interestingly, cell non-autonomous polarization offers much more robust polarity patterning outcomes. So the next thing is, where do we take this model? So PCP is an important mechanism that guides various cell behaviors during tissue morphogenesis. So using CompuCell3D, we can easily model PCP-driven cell behaviors like cell elongation, which was done using the focal point plasticity plugin in CompuCell3D. Then we could do PCP-mediated cell migration, where that would guide the movement of cells, which was done using external potential plugin. And finally, mediolateral intercalation in gastrulation or primitive streak formation, where you have these cells which are planar polarized along the anterior-posterior tissue axis, and then they pull on each other along that axis. and undergo convergent extension along the anterior-posterior axis over time. So PCP guides the convergence axis. So that's with the work, and I would like to thank my advisor, Dr. Giulio Belmonte, and Dr. Eric Brooks, and all members of Belmonte Group. So this is basically the story, and now I'm going to shift gears and going to start talking about the implementation aspects of the model. So before that, is everyone able to download the code that that was shared? So everyone can download it and unzip and load it into CompuCell3D player and open it in Twedit as well. Cool. So as you can see in the simulation, each cell has 4 domains, right? So you have cytoplasm, proximal, distal, and lateral. So I will first talk about how did we decide on the contact energies between the different domains, and then we'll run the code and I'll explain each section of it. So as I said before, the goal is to have the proximal and distal domains across cell-cell boundaries to have high affinity. So the first natural thing to do is set the external contact energy between the proximal and distal domains to be lower and everything else to be higher. Now, if you look at cytoplasm, you don't want cytoplasm to interact with the medium or the cytoplasm of other cells. So you set the external contact energy for cytoplasm with every other domain to be really high. And then the proximal distal energies are the same, but it's higher than proximal distal energies. Similarly for lateral and proximal lateral and distal lateral. So the way we arrived at these final set of energies is by extensive sensitivity analysis for each of the parameter combinations. But essentially, the key idea is that as long as you have proximal distal energies to be lower compared to every other set, it works. And now coming to internal contact energies, we are looking at 2 possible cases. As I explained before, cell non-autonomous polarization and cell autonomous. So cell non-autonomous polarization requires a cell, if it's isolated and if you prevent it from interacting with its neighbors, it should have the compartments mixed inside its membrane, right? So you would still have the cytoplasm to have higher energies with everything external. So basically, if I have one single cell surrounded by medium, you would still have cytoplasm and medium to have a higher energy. But the only difference is now within a cell, you don't want the proximal, distal, and lateral compartments to be spatially segregated. You want them to be mixed. So what I do is that I set the contact energies between every other compartment to be -1, the internal contact energies. So this would make sure that in isolation, a single cell would have the compartments mixed in their membrane. Now, the second scenario is cell autonomous polarization, where when you have a single cell and it's surrounded by medium, and you want the proximal distal compartments to be spatially segregated because the cells can do it autonomously. So the way you implement it is by setting the internal contact energy between the proximal distal compartments to be high, and then you can see that it's spatially segregated. So we'll run this simulation, but before that, I'm sure if you have run the simulation as it is, you must have been able to see 3 windows. And initially, you can see that you have 5 cells along both X and Y directions. And within each cell, the compartments are really mixed. And now as I evolve the time, you can see that the compartments start sorting out within each cell. Now here I have the orientation vectors, which are basically pointing from the proximal to distal direction, basically the pink to green direction. And here I'm plotting the global polarization. I will come to how we are calculating this later, but as of now, just focus on the fact that the global polarization, as I mentioned earlier, is calculated. It has values between 0 and 1. So 1 would mean perfect alignment, and 0 is like a perfectly disordered system. So you can see that how the global polarization fluctuates as the system is self-organizing. So were you able to run the simulation, everyone? Okay, cool. So now let me go to the code first and show a brief. Yeah.
Is
So
it possible to, yeah, I was going to say that whatever that dialog box is that you've got your cursor on right now keeps
mhm.
blocking different places on your screen.
Okay, I was like, how do I view the chat without? Okay, or can somebody read it to me if there are questions? Um,
I
okay.
would recommend if people have questions, just go ahead and ask when if you have any problems or anything. I can monitor chat.
Okay, thank you so much. So the way the code works is so far you must have seen you have the XML file, then you have the main Python file and a steppable file. But I have used only the main Python and steppable file. So whatever you write in the XML file, you can do that in the main Python file as well. So starting here, so I define the typical diameter of the cell and how many cells do you want in your lattice and the size of the lattice. And then I have the Potts parameters like temperature, neighbor order, and total time. And then the so for the volume constraint of the cells, I have the parameter that determines the strength of the volume constraint, lambda volume, and targets volume of the cells. And most importantly, so since we are assigning certain fraction of the cell volume to be proximal domain, the other set to be distal, lateral, etc. So the way I do that is by assigning 15% of the total volume of the cell to be proximal, 15% to be distal, and half of it to be cytoplasm, and everything else to be the lateral domain. So I define that here. And then I define the external contact energies between the different compartments, basically between cells, as well as the internal contact energies. So then so you have all the Potts parameters defined here, and then the cell types in the simulation, cytoplasm, proximal, distal, and lateral, and the contact energies. I also have the volume local flex plugin. And now is the important part. So the first question is, how am I drawing the initial conditions for the simulation? So I'm not using uniform initializer. I'm drawing it at the Python level using the steppable file. So here you can see three different initial conditions that we will be looking at in this session. So right now, what we are using is this periodic boundary conditions initial condition. So I would first want everyone to comment that and start with the single cell scenario. So uncomment that. And then you can run the simulation. Is everyone able to see this? Um, okay, so if I run this, you can ignore the global polarization and the orientation vector. But if you focus on the single cell, you can see that the compartments are not always spatially segregated. It is it's kind of mixed. For instance, if I pause here, it's okay, but then I run and you can see that it's mixed, the proximal and distal compartments. So you keep doing it, you can see that it's mixed. So this is because the internal contact energies that I'm using are -1 for all the domains within each cell. Now, what I want you all to do is change this. So you set the proximal distal contact energy to be really high and the other set to be 1 positive, and see how would the simulation look like. Do you see that the proximal distal compartments are spatially segregated at all times? Was everyone able to run the simulation and see? So you can see that even as time progresses, the proximal and distal compartments stay spatially segregated at all times. So that's the cell autonomous version of the model. So now I want everyone to put back the internal contact energies to be -1. Was everyone able to see the spatial segregation of proximal distal compartments when you change the internal contact energies? Can you do this? Can you create a new project, say new CompuCell3D project, and do just Python only, and then continue, and then copy paste the whole thing as I'm doing right now. Like this, but make sure that when you do this, you set the name of the simulation to be PCP _simple.cc3d. Yeah, yeah, you could do that as well if you're on Linux. Okay, cool. So let me continue. So next, so that was the overview of the main Python file. So now I have the Steppable file where I have all the steppables that I have been using. So we have the 3 initial conditions, one for a single cell surrounded by medium. I'll explain how each of these work. Then I have one for the periodic boundary conditions and another for left boundary signal and a metric calculation steppable and how to plot the metric in CompuCell3D, and finally visualizing the orientation fields. So first, if we go to drawing the initial conditions, basically placing the single cell on the lattice, let me open this. So here I have listed the parameters that I'll be using for initializing this. So I have the cell diameter. I need to set the target volume and lambda volume for the cell. And as I mentioned before, how much of a cell should be proximal, how much should be distal. So the proportion of the various compartments within the cell volume. So now the way I initialize the cell is by starting with by creating a new cell type called cyto, for instance. And then I want to start. So if this is my lattice, I want to draw the cell, say, at 1 cell diameter apart from the origin. So this is my starting point. Self.cd is basically the cell diameter. And then I want the cells to be of, say, 12 pixels. That's my cell diameter. So I do a for loop and then along x direction as well as y direction, and then place the cell at each of those pixels. After that, I set the target volume for this particular cell to be cell diameters squared and the lambda volume. So is this part clear for everyone? Next, I'll be talking about how to pixelate the cell, how do we assign the compartments to different regions of the cell, and how do I randomly distribute it within the cell. So if this part is clear, then I can proceed to that. Is this okay for everyone? Cool. Now, okay. Now is the important part. So how do we assign some part of the cell's pixels to a certain compartment, the rest to the other compartment, etc. So before we do that, we need to have a list of the pixels that make up this cell, right? So for that, I have this function. What it does is, so you could do this using the pixel tracker plugin as well in CompuCell3D. But the thing is, for this simulation, you need this only at the very beginning, at the initial time step. So if I use pixel tracker, then it would be used at every time step. So that would slow down the simulation. So, which is why I wrote a custom function that would do that only at the beginning. So the way it works is, first, I create an empty dictionary, and then I loop over all the cells in my lattice. So here I have just one cell, but in the general case, then for the cell ID, I would make a list, and then I would go over all the pixels in the lattice and identify and grab which cell belongs to that pixel. And then if that cell exists, then I add it to this dictionary for that particular cell ID. So in this fashion, I get the list of pixels that make up that particular cell. Make sense? So after this, so if you have any questions, please interrupt me. Okay. So after this, what I do is that, so at the beginning, what did we do? We drew a cell at, say, this particular region of the lattice, which was this part. And then we assigned a target volume and lambda volume for that particular cell. And then I called that custom function that I wrote in the previous slide. And what it does is it creates this dictionary with, for each cell ID, you have the list of pixels that make up that cell. And then finally, what I do is I want to pixelate the cell, basically assign the set of certain fraction to be proximal, the other to be distal, etc. So if I have many cells, so I just go loop over all the cells in the simulation. So here I just have one side to cell. So I do that, and then I call the pixelate function. Now, how does the pixelate function work? So first, we have to create proximal, distal, and lateral compartments, right? We already have cytoplasm. That's the original identity of the cell. And then I make an empty list, which would basically grab all the pixels that's making up that particular cell that we have, which is what I'm doing here from the dictionary that we created before. And then what I do is I shuffle the pixel list. So it's not sequentially ordered anymore. And then what I do is I grab each pixel from that list, and then I generate a random number between 0 and 1. And if it's less than, say, 15% was my fraction of proximal pixels I want. So if it's less than that, then I assign that pixel to be a proximal domain. Similarly, if it's less than this, I assign it to be a distal domain, like that. So in this fashion, you're randomly assigning each pixel to be a different domain by keeping the relative the proportion. So 15% of the cell volume would eventually be comprised of proximal cells, 15 would be distal pixels, etc. Does that make sense? Cool. So then the most important thing is now, as Julie was discussing this morning, how do we make sure that all these compartments are belonging to this one particular cell? We have to first assign a target volume for each of these, which I do here. So 15% to be this, blah, blah, blah. And then the most important step is to assign the cluster ID for each of the compartments. And assign it to the same cluster as the initial cell that we had, which is cytoplasm. So once we do this, we get an initial condition, which we saw at the very beginning, the time step, where you have a single cell and the compartments are uniformly mixed inside the cell. And then just by using the contact energy preferences, it spatially segregate within each cell. So this is the basic idea. Once this is clear, I would go ahead and describe how I generated the two different boundary conditions. Okay, I think I'll go ahead. If you have questions, feel free to post it in the chat. Okay, so the next thing is the periodic boundary conditions. The idea is the same. So now we learned how to pixelate a single cell. So now we are going to draw we're going to start with an initial condition where I have cells arranged in rows like this, and then I would pixelate them, and eventually the system would self-organize into a configuration like this. So first, what do we need to do? We have to comment the initial condition steppable that would draw a single cell in your main Python file, and then go to the second boundary condition where we're using periodic boundary conditions and save it. And then if you start playing, you can see that I have 5 by 5 cells. You can change the number of cells if you want by changing it here at the beginning of the main Python file. So you can change the number of cells along both X and Y directions if you want. Now, the next question is, how do I place cells within the lattice fully packed? So you have the cell diameter and you have the dimensions of the lattice as well. The way you do it is again by so if you watch closely, you have an offset here because I want the cells to be hexagonally packed inside the lattice, which is why I start with an offset. So this is how we want to draw the cells. So this part of the periodic condition steppable would do that. So we start with an offset YY, a vertical offset, which starts at half of the cell diameter. And then I loop over the whole X dimensions as well as Y dimensions. And then I create a new cell, cytoplasm, and assign target volume and lambda volume for that cell. And then I assign each pixel to be a cell. So this Y0 plus Y plus YY is the start position plus how far into the block we are plus how much the column is shifted, as you see here. And this part is to make sure that, so remember we have periodic boundary conditions along both the directions. So if the column shift pushes past, then you want to wrap it around. So that's what this part does. And once you have the cells, it's simple as before, like we did for the single cell. You would assign target volume and lambda volume. Then you would get the dictionary of pixels corresponding to each cell. And then you would pixelate each cell, exactly like we did for the single cell. So that would give you the periodic boundary conditions. Is that clear? So if that's clear, you can go ahead and play the periodic boundary condition simulation. And you can see that there is no preferred direction of alignment for the whole tissue because we are not telling the cells which way to align, right? So it can align from along any direction, the whole tissue. And you can see how the global polarization evolves over time. So if we wait sufficiently longer, then you'll be able to see that the global polarization right now it's at about 0.5. But if you wait longer, you'll be able to see that it reaches close to 1. So you can wait and see. So here you can see the orientation field vectors. Is everyone able to see that? If not, you can just click here. So you have two different views, cell field and the orientation. So you can switch between both. So everyone was able to run this and see the global polarization increasing over time. So you can see that it's right now increasing over time. You can see the orientation field vectors are all aligned along mostly this direction. Okay, now the question is, so this particular scenario, so as I said, there is no preferred direction of alignment for the whole tissue, right? So now how do we force the tissue to align along a specific direction? Say, for instance, left to right, proximal to distal direction. So the way I will do this is by introducing a boundary orienting signal at the left side of the tissue. So this would force the first column of cells to align from proximal to distal direction, and then it should propagate further away from the boundary. So to do this, I want everyone to uncomment this initial conditions left boundary in your main Python file. So you would uncomment this, and then you should be able to see the initial condition where you have two columns of pixels are now the distal compartment, and then you have everything else the same as before. So you have periodic boundary conditions along y direction, and you have a boundary orienting signal. So the way this is implemented is by, so you want the first two columns of pixels to be of the cell type distal. So that would force the first column of cells to align along proximal to distal direction. So the way you do it is by creating uh, a new cell type distal, and I call it signal. And then I loop over all the pixels along y direction, and I set for x equal to 0 and for x equal to 1, I set it to be this cell type which I created, that is distal. And the important part is you want this boundary to be fixed. So, which means I want it to not move or fluctuate. So, I would set the lambda volume to be really high. Okay. So you set the lambda volume to be really high so that the boundary is fixed. And everything else is the same, except that now instead of starting at 0 to draw your cells, you would start after the boundary, basically from 2. And the method in which you place the cells and you pixelate them is exactly the same as before. So if you run the simulation, you can go ahead and run it. If you run the simulation, you can see that as time progresses, the cells would try to align along proximal to distal, left to right direction. Do you see that? So you can see that clearly the global polarization is increasing over time and the cells are aligning. So now we'll come to the first activity where I would want you to create another boundary on the right side of the tissue, and the type of this boundary should be proximal, and I want you to see what happens to the alignment in such a scenario. So before you start doing it, I would recommend you to first change the dimension. So if you go to line number 13, you comment that and you comment line 13 and uncomment the line below to adjust for the 2 pixels that you would be adding on the right side. But can you go ahead and add a boundary on the right side, and it should be of the type proximal, and see how that would change the alignment of the tissue. Okay, good question. So you want it to be of proximal type, right? So what you do is basically create the signal to be of type proximal. Isn't that what you asked? Cool. Yeah, and then you would basically set it at the last 2 pixels, right? So that's why I set x to be self.dim.x, which is the maximum dimensions along x-axis, -1 and -2, and then everything else is the same. So that would give you this. So if you run it, I guess you should be able to see that the results are still the same, because the distal domains, the green domains, would still have proximal on the other side. So you would still have every cell to be aligned along proximal to distal direction. Probably you would see that the cells achieve this much faster than the previous scenario where you had just one distal boundary. Now, the next thing I would want you to do is switch this proximal to be of distal. So you have 2 distal boundaries on both the sides and see what happens. So essentially it's the same as before, except that you make it to be of distal type and then you add these things as well. So what do you get as outcome? It's like the cells here and here are pointing along opposite directions. So the cells here eventually the point up or down, as you see. Cool. So now the third thing that I want you to try is so here we had boundary signal, right? Now, instead of that, if we remove that and if we go back to the previous initial condition where we had periodic boundary conditions along both the directions. So, and if I have a cell at the center, which is acting like a source, instead of a boundary, now you have an orienting signal at the center of the tissue, which is of the type, say, distal. So what would happen to the alignment of the cells near it? So to do this, you will have to, okay, let me guide you through this. So you will first have to go back to the previous initial conditions where you had periodic boundary, because as you see, we are using the periodic boundary initial conditions, and then replacing the cell at the center to be of distal type, and you don't want this cell to be pixelated. So what would you do to do this? If you have any ideas on how to do this. So the first question is about how would you choose this particular cell, right? So you don't want to pixelate all the cells. You want to pixelate every cell except this one. So the question is, how would you choose this? So you could choose this based on where it is, the center of mass of this cell. So for instance, it's at roughly halfway through the lattice dimensions. So you could choose it based on that. And then not pixelate it. So can everyone try for once? And then if you're facing trouble, I'll show you how to do it exactly. Also, one more thing, since you're using periodic boundary conditions, you also have to switch back to the dimensions of the lattice like we had before. So you need to switch it back to this, and then should I go ahead and show how to do it, or are you still working on it? If you could let me know in the chat. So let me just, so basically, in the periodic boundary conditions, we were pixelating every single cell, right? So as I said before, we don't want to do that for this particular cell. So first, what do? I loop over all the cells in the tissue, so all the cytocells, and then if my cell is at the center, so the way I check that is by checking the center of mass of this particular cell. So if its X center of mass is within say half of the lattice dimensions, plus minus 5 pixels, and similarly along the y direction, plus minus 5 pixels at half of y lattice dimensions. And then if that is the case, then what do? I switch the type of that particular cytocell to be of distal. And then I don't want to pixelate it. So I would pixelate only the other set of cells. Does that make sense? So you can try this. Yes, you have to comment out the boundary signal initial conditions. Yes. So yeah, so you should be able to see a scenario where if you wait longer, you should be able to see that the proximal domains would come together near this source, and hence all the cells would point away from the source. I hope you should have been able to see that. So ultimately, the point is by controlling the domain types, the method is so powerful that you can use it as orienting signals, and just by changing the contact energy preferences of the domains within each cell and between the cells, you can study this polarization behavior of cells. And now I'm going to switch gears a little bit and talk about how do I calculate the metrics that we have been looking at. So for instance, we saw that global polarization increases over time, right? So how are we calculating this at the steppable level? So I have two steppables for this, cell polarization and graph global polarization. So this would plot the global polarization. So first, let me look at the cell polarization steppable. So the way it works is, so essentially what we are trying to look at is the polarity vector for each cell in the lattice. So this is basically the vector that points from the center of mass of the proximal to the distal compartment. So we will first get that, and then I'll get to the next part later. So first, how do we get the polarity vector for each cell? So this is done in the cell polarization steppable. So first, we go over all the cells in the lattice. So I go by the cell type cyto. And then I look at all the compartments that make up the cell cluster that this cyto belongs to. So I loop over all the cell compartments, and then if I find a cell compartment to be proximal, I store the center of mass, both X and Y center of mass of that compartment. Similarly, if this compartment is of distal type, I store the X and Y center of mass of the distal compartment in these variables, so xp/yp and xd/yd, and I also keep track of if that compartment exists or not. So I had initialized these variables here. So now if OK equals 2, meaning if both the compartments are found, only then I would proceed to finding the polarity vector. So then I find the difference in the center of mass of X and Y coordinates. And then remember, you have periodic boundary conditions. So you have to adjust for that, which I'm doing here. You can ignore it for now. And then I normalize this distance that I calculated. And finally, the important thing is, I don't know if this is being introduced yet in the workshop, but for each cell in CompuCell3D, there is a by default dictionary, Python dictionary associated with it. So we can add attributes to this dictionary and use it for many things. So here what I'm doing is I'm using that dictionary, I'm adding an attribute which corresponds to the polarity vector of that particular cell. So for each cytocell, I am adding this attribute, the unit vectors along x and y direction, as you see here, and another attribute called polarization, which is essentially a measure of how much spatially segregated the two proximal and distal domains are. So you can ignore this, but importantly, the idea is that you can add attributes into the cell dictionary, and we will use this to calculate global polarization. Is that clear? Now, the question is, we are interested in calculating the tissue scale polarity order, right? So what we have right now is we saved the polarity vector corresponding to each cell, the x and y unit vectors of that in the dictionary for that cell. Now we will use this Steppables, basically graph global polarization, to calculate this global polarization. So how do we do that? Essentially, it's like this question of how does the polarity vectors of each cell agree with each other, right? So you can see that if I have vectors like this, then so we saved this unit vector in these dictionary attributes corresponding to each cell. So now what we will do is we'll sum all these vectors tip to tail, and then we will take the magnitude of that and then divide it by how many cells I have. So that's exactly what I'm doing here. So I again go over all the cells in the lattice and I keep a track of how many cells are there. And then I'm adding the x component and the y component separately, is essentially this part where I'm summing up the vectors. So this is good because we saved this as a different attribute in the cell dictionary. So we can easily access that. So and then you find, we find the magnitude, and using that you normalize it by the total number of cells, which is n here, which we calculated here, and that would give you the global polarization phi. Now, how do we visualize this? Yeah, so we can visualize this using using this in CompuCell3D. So I have a plotting window. So we initialize it at the start function. I'm not sure if this is introduced yet, but okay, so you initialize it. So you can add the title and the XY axis and everything. And then once we find phi, as I explained in the previous slide, you can just plot it in this fashion. So you just add the data point, which is basically time, MCS versus phi. One additional thing that you could do is also, if you want, you can write these values into a text file or CSV file at the end of the simulation. So that's why I'm saving these values into lists, as you see here, M and phi matrix. And oops, I think I don't have, okay, let me go back to the code. So in the code, if you scroll all the way down to graph global polarization, you can see that, okay, here I'm initializing the plot, I'm calculating phi, and then I'm storing these in matrices. So I have time versus phi, and at the end of simulation, I can save this into a text file, for instance, globalpol.txt, which I can use later for further analysis and stuff. So that's one thing you could do. And finally, you could also, how are we plotting the orientation field vectors? So you do this using orientation field Steppables. So you create the vector field, and again, using the same dictionary that we created earlier, the attribute that we created earlier. So if celldict.pol is not equal to 0, meaning, remember I mentioned that this particular attribute actually measures if the two compartments are spatially segregated at all or not. So if it is spatially segregated, only then do you want to plot the polarity vectors. So you give the unit vectors and it plots it. So there is a negative sign here because it just depends on the way I defined the vectors. I think I defined it the other way, proximal minus distal, which is why you have this negative sign. So if you flip it, you can remove this negative sign as well. So that would ultimately produce the orientation field vectors. So that's pretty much it with the simulation, but the simulation code for the model with proliferation is uploaded on my GitHub. So if you're interested, you can check that. So it's a bit more complicated, which is why I didn't present it here. But if you have any questions, you can let me know. No, it's about do you need morphogens for the cells to polarize? Like, I guess it's organism dependent. But the thing is, we still don't have an understanding of if at all cells are using morphogens for this. Okay, so what role does the polarization plays in morphogenesis? If you come to the context of PCP, it's really important for convergent extension, for instance, like that determines the body axis elongation in vertebrates. So PCP provides the convergence axis for the convergent extension. And what role do morphogens play? No, I'm not saying morphogens don't play a role in morphogenesis. I'm just saying that the question is if morphogens are required for polarization at all. It's still not very well understood. Of course, morphogens do play a role, but yeah, the question is it absolutely required for planar cell polarity? Good question. We haven't done that, but I did try changing the size of all the cells and looking at if I still get the same result. And as expected, it depends on the neighbor order that we use in the simulations. So think about it. The problem is if I make the cells to be really large, uh, I'm in. So what the ability of the proteins or the domains that I have to diffuse across the cell is being disrupted, right? So I have to change the neighbor order as I change the cell sizes. But I actually did not vary the cell sizes in such a way that one is bigger than the other. But the other thing is, in this implementation that I showed, we were drawing cells as like perfectly packed in the lattice. But in contrast, in the simulations that I used for the paper, we don't do that. What we do is we place the cells as random seeds in the lattice and then let them grow and pack the lattice. So yeah, so there is some variability there, but again, it's not it's not like I have some small cells and big. But I think that would be an interesting thing to look at because it would affect the way the cells are packed in the lattice, and that certainly would have an effect on global polarization.
Well, if there are no other questions, let's thank Abhisha for her talk and her demo. It was great. Thank you so much.
Thank you, everyone. Thank you for the opportunity as well.